For the longest of time, we thought that the genetic changes between your genome and my genome were essentially only in single nucleotide changes. What structural genetic variation is parts of the genome that are lost or gained or rearranged. One analogy that you can use is like a photograph where pixels that are deleted, that are missing, actually hide something important, some image that's really important underneath. And then once you realize it's there, you get a more complete understanding, a complete visual. I led one of the two teams that made the initial discovery of structural genomic variation in the human genome back in 2004. When I came to the JAX laboratory, I obviously continued my research and helped to recruit others that were interested in structural genomic variation, such as Christine Beck.
I like knowing how things work, and that combination of imagination and coming up with good questions and then exploring them through hypothesis-driven research was really what led me to get a PhD and follow down that path and be here.
You have a Y on the left and an S on the right of the two that you have joined there. It's a Y.
It's a Y, then an S. What the last few years has really brought to bear is the fact that we can now assemble a contiguous genome. So it was only just a few short years ago that we actually had the first telomere to telomere representation of a haploid human genome. So once you have these kind of larger duplications in genomes, then those regions are more labile for rearrangement. However, getting those regions assembled is difficult because with shorter reads or smaller pieces of a genome, you don't know if a block of sequence comes from region A or region B, or if instead of a duplication, if it's a triplicated region or a quadruplicated region, you'd have to basically choose out of a hat which locus that sequence came from. So with these beautiful contiguous assemblies that stretch for hundreds of megabases in some cases, or tens of megabases in some regions, you can actually thread through these segmental duplications and through these complex genomic rearrangements. And then because your assembly can get through those regions, you can then call differences in one person versus a reference genome. Now we can resolve dozens of genomes. So in this study, we sequenced 65 individuals, and we sequenced all 65 of them from nearly telomere to telomere, and also looked specifically at complex genomic rearrangements between individuals. It's hard to explain how much the technological advancement really enables the knowledge advancement in genomics and genetics.
We didn't have a great tool for getting structural variants from the assemblies that could handle the errors that we get when we take someone's genome and we line it up to a reference and look for the differences. There's a lot of noise. There's a lot of errors that happen in that. So I wrote a program to pull out variants that we weren't able to see with very similar or with long reads and assemblies just a few years ago. And in particular, the complex structural rearrangements. Yeah, so this would be our reference genome, and we have these gene annotations. In this gray bar represents someone's DNA that we've sequenced and assembled and put together. And when we line it up to the reference genome, we find that it lines up to here, and then it jumps back and makes an extra copy of this piece from here to here. And then it makes a second jump here, creating this complex structural rearrangement. And so what's nice about the technology that we're working with today is that we can see the big stuff and we can see the small stuff all together into one picture. And that could make all the difference between correctly deciding if somebody has an increased risk of cancers or not. The level of information that we're able to get today is really setting the stage to change how we care for people in the future.
Part of the reason why I love science is because it's basically a puzzle. And part of the reason why I love what I specifically do in the context of genetics is because it's taking a human genome. And now that we have that resolution, you can put all those puzzle pieces together and actually get an adequate view, a really clear view of the differences between individuals. That's why I think that what we have as data in hand are really beautiful, because I'm interested in not just how we vary, but also the processes that got us there. And without those pieces of the genome all together in contiguous space and beautifully resolved with Pete's methods, we don't have a good picture of that.